We're going to take a look at some of the internal structures that we find inside bacterial cells. So the bacterial cells do have a cytoplasm, just like eukaryotic cells do. So this is the gel-like material made mostly of water that's located inside the plasma membrane. And then bacterial cells also have a cytoskeleton, which is very similar to the cytoskeleton we find in eukaryotic cells. So there are 3 different proteins that make up the cytoskeleton in bacterial cells. They're not exactly the same as microtubules, microfilaments, and intermediate filaments that we find in eukaryotic cells, but they're very similar. And they're going to serve the same functions as we find in eukaryotic cells. So they're going to have a role in cell division and also in determining the shape of the cell. Here are some pictures of the bacterial cytoskeleton. And if you notice the fluorescent colors in these images, you know that these were taken with a fluorescent microscope. And what they did is they used antibodies that were specific to binding to certain parts of the cytoskeleton and then put different dyes on those antibodies. So then we're able to see the different parts of the cytoskeleton. So you can see how they're definitely involved in cell shape. And also these bacteria are in the process of dividing, so you can see that they're involved in the middle there, where those 2 cells would then split apart. And on the bottom picture, if you remember the shapes of the bacterial cells, this is the Vibrio shape. So remember they're spiral, they're kind of like a curved comma shape. And what gives them that shape is the cytoskeleton that's inside that cytoplasm. And then this is just another picture of the cytoskeleton inside these bacterial cells. The nucleoid of a bacterial cell is the area in the cytoplasm where the DNA is located. There is no membrane that surrounds the chromosome here, so there is no nucleus. So a nucleus is not the same as a nucleoid. The nucleoid is just the area of the cytoplasm where the chromosome is floating around. Bacterial cells only have 1 chromosome, and the chromosome shape is circular. So that's really different from eukaryotic cells, which are going to have 2 or more chromosomes, and their chromosomes would be linear instead. And because bacteria only have 1 chromosome, they're going to be haploid. Remember, haploid means that you only have 1 copy of each gene, whereas we're diploid because we've got 2 chromosomes that are the same, 1 from mom, 1 from dad, so we're going to have 2 copies of each gene. But because the bacteria only have 1 chromosome, they're haploid and only have 1 copy of each gene. And this is just a picture of the nucleoid. You can see the DNA in the top that's been stained. And just that area of the cytoplasm where it's floating around, that's called the nucleoid. So there's no membrane around that. And this bacteria is getting ready to finish dividing here. And then this E. coli cell on the bottom, they've actually unraveled the chromosome and spread it out outside the cell. So that's a lot of DNA to stuff into that E. coli cell. So how the chromosome is going to fit into that bacterial cell is we need to package it up and coil it. So remember in eukaryotic cells, we also package up our DNA and wind it around some little spools called histone proteins. We're also going to be using very similar protein spools to wound the DNA around in the prokaryotic cells as well. But they're not the histone protein. They're very similar though, similar function, but they're not histones. Plasmids are pieces of DNA that are extrachromosomal. So that just means that you're going to find this plasmid outside of the chromosome. So a lot of the bacteria and archaea do have plasmids, but not all of them do. And for the most part, we do not find plasmids in eukaryotic cells. There are a few yeasts that have them, but in general, eukaryotes don't have plasmids. The plasmids don't have to be there. The bacteria can live without them, but the plasmids usually contain genes that give the bacteria an advantage to survive in the environment. And one of the reasons that we're concerned about plasmids is that this is where a lot of our antibiotic resistance genes are located in bacterial cells. When a bacterial cell divides, it not only replicates its chromosome to pass to its offspring, it also will replicate its plasmids. And so those antibiotic resistance genes that are on the plasmids will then get passed to the offspring as well. And in this top picture, you can see that this bacterial cell actually has 3 plasmids. And sometimes some bacteria will have 1 plasmid, some will have several, and then there's others that don't have any plasmids, so it just depends. And then the bottom picture is just showing you that sometimes these plasmids can cut the DNA and insert themselves and make them part of the chromosome. And so when the bacteria divides and replicates its chromosome, it also replicates that plasmid as well, and then those antibiotic resistance genes get passed on to the offspring as well. Remember that ribosomes are protein-making machines, so their job is to make proteins. So they're going to translate the messenger RNA and read the code and figure out what amino acid to put in what order. So they're going to be made up of proteins and ribosomal RNA. And the ribosomal RNA that's inside the ribosome is actually an enzyme, and it's going to catalyze the formation of the peptide bonds. And the ribosomes consist of 2 subunits. There's a small ribosomal subunit and a large ribosomal subunit. So these 2 pieces are separate until they come together and bind to the messenger RNA to do translation, and then they make the complete ribosome. We do have different sizes of ribosomes in prokaryotes versus eukaryotes. So bacteria and archaea have 70S ribosomes, and eukaryotes have 80S ribosomes. The S is just a unit of measurement. When you centrifuge the ribosomes, the bacteria and archaeal ribosomes are smaller, and so they're going to be at a different level than the eukaryotic ribosomes, which are heavier. And so that S just stands for Svedberg unit. You don't need to know that, but it's just a unit of measurement they use when they centrifuge these ribosomes. So the reason it's important to know the difference between the prokaryotic and eukaryotic ribosomes is that we do have a group of antibiotics that will act on the 70S ribosomes, which are found in the bacteria, and that's how they kill them. So the bacteria aren't able to make their proteins anymore. And these antibiotics don't affect our cells because we have 80S ribosomes instead. We also find inside some of the bacteria inclusions. Inclusions are used for a number of different things. They could be used to store things. They're used for doing chemical reactions, some sort of metabolic reaction. And then they can also be used to position the cell in water. And what's interesting about these inclusions is that some of them are enclosed by a membrane. And the membrane can vary in composition. Some of the membranes are just made out of proteins and that's it. And then others will contain some lipids and proteins together. And so you can kind of see where these inclusions may have evolved into organelles in eukaryotic cells. So storage inclusions are going to be storing nutrients, energy stores, things like that. So you don't need to memorize this list, but just know what a storage inclusion is. So some of these will be storing glycogen and energy stores. Some of them will store carbon, so the bacteria will have an easy way to make organic molecules with the carbon. Some of them store phosphate, which remember phosphate is important to make nucleotides and ATP. And then some of them will store sulfur. And this is just an example of a storage inclusion down here, which is very interesting. The membrane that's surrounding it has some phospholipids and also some proteins in it. So you can see where maybe organelles might have evolved from these storage inclusions. Other inclusions are used for metabolic reactions. So an example here are carboxysomes. So these are found in bacteria that are doing photosynthesis and fixing carbon dioxide. So that would be, for example, cyanobacteria. And remember that when you fix carbon dioxide, you're going to take carbon dioxide gas from the air and make it part of an organic molecule. And what's inside of these carboxysomes is the enzyme Rubisco. And you remember learning that before? Because remember, Rubisco is used in the Calvin cycle to fix carbon dioxide, so we're going to say that Rubisco is used for photosynthesis. Okay, and these carboxysomes, they actually have a protein membrane that surrounds them, and the Rubisco would be located inside of there. We also have some inclusions that are used to position bacteria that are living in the water. So we have gas vacuoles. These are found in aquatic bacteria and archaea, and these have a protein shell, and inside they have gas stored. So they can put more gas in there if they need to float higher in the water, or take some of the gas out if they want to sink lower to where more food is. So that's pretty cool. And then we also find in aquatic bacteria these magnetosomes. So they have little inclusions that contain iron oxide, which acts as a compass. So they can actually detect the magnetic field. And so when they're swimming around, they're able to tell like which direction is north, which is really cool too.